Hey guys, how are you today?
Good idea.
Wonderful, thank you. All right, need a few more minutes. Anybody do anything fun over the weekend?
Nope.
I did. I went on a whale watching boat. That was really fun. There were dozens of whales. It was very interesting. I've never seen a whale in the wild before. I've seen them, of course, in
Sorry, I'm late.
oh, that's okay, Zion. That's okay. You're just like 1 minute late. So that's Luke, I went in Boston out into the Atlantic from Boston. We sailed out for about an hour, maybe an hour and a half before we started seeing whales. And then they were everywhere. There was even a young one that was with its mother. That was kind of cool. They're not up for very long. You have to really be watching, but when they turn and dive down in the tail, they were humpback whales. So they had that beautiful tail as it went down. It was very cool. All right, let's go ahead and get started. Nobody told me they weren't going to be here. It is helpful if you guys let me know if you're not coming to class. Not only do you get an excused absence, but then I know not to wait. All right, so let's start here. Okay, you can see that where it says photosynthesis review. So we're going to start out today by talking about the bromothymol blue lab that we did last week. So today I want to talk about the results that we got. So if you did the lab and filled out the form with yours, hopefully you have it with you. If you didn't, you can look on with mine, but either way, I wanted you to use the lab sheet and turn that, or you don't have to submit that. I just wanted you to bring it today. So we did this lab together. Did anybody have anything unusual happen? Anything that was unexpected? All right, let's take a look here. Remember, this was our hypothesis. So our hypothesis was if a plant is placed in a bromothymol blue solution and placed in the light, then the color of the solution will change from yellowish green to blue. So what was our independent variable in this experiment?
The test tube without the plant.
Possibly. Let me think here. The test tube without the plant. That would have been the control. Yeah, good guess, but that's the control. So remember, independent variable is the one you change. I'll sing it if you want. Independent variable is the one you change. Independent variable is the one you change. Dependent one is what you measure. Controls stay the same.
Wouldn't it be the water or the bromothymol blue?
Sorry, Abigail, say it again.
Would it be like what's with the bromothymol blue? Or no? There's too many things that change.
I know. The bromothymol blue was our indicator. That was just telling us whether there was carbon dioxide or not in the solution. So the plant and the light, remember your independent variable is the if part of this. If we do this, and then the then part is the dependent variable. So if we put the plant in if the plant is placed in the bromothymol blue and placed in the light. So the plant in the bromothymol blue with the light is the whole independent variable. And then the color of the solution will change from yellowish green to blue. That is our dependent variable. All right. We also talked about how we could do it because if we added because the plant will give off oxygen and take in carbon dioxide as it performs photosynthesis. You don't have to do the because for my class, but I wanted you to be aware of it so that, in other classes, if you get that. Now, we followed these procedures in class, but then you guys did number 8 on your own, either with your own experiment that you did or the pictures that I sent you. So I'm going to show you the pictures that I sent you, and then I'll show you how I completed my chart. So take a look at your own chart, and if you get something different, let's talk about it. So the initial color for all three was this yellowish green. So that was our initial color. That was, remember, it was when we had the bromothymol blue in the water and I blew into it and it turned into that yellowish green color. So that was the initial. Then at 15 minutes, this is what we saw. So you can see there's no change. It's still this kind of yellowish greenish color. It looks a lot more yellow green in real life than it did here. It almost looks clear in this, but it was still yellowish green at 15 minutes. And then again at 30 minutes, it was still the yellowish green color. And then again at an hour, nothing had really changed at an hour. Now, by the time we get to about 8 hours, then, we saw that this experimental one became a little bit pale blue. So there was a little bit of a difference in the one that was in the experimental light, but it took some time for photosynthesis to take that carbon dioxide out of the air or out of the water. And then at 24 hours, there was a big difference. So you can see right there, it is a darker blue, a brighter blue. So the control didn't change and the one in the dark didn't change, which we were expecting. We were expecting them not to change. Bethany.
How should we dispose of the solution? Can we just pour it in the sink? What do we do
You
with
just
it?
pour it right down the sink. It's perfectly safe.
Okay.
Yep.
Okay, just want to make sure.
Yeah, thank you. I'll make sure to put that in the instructions for future classes. All right, now let's talk about the analysis, because I also wanted you guys to do the analysis section of this, to write those down. So how did you answer the top one? Describe what happened to the color of the bromothymol blue in the test tubes with and without the plant under light conditions. Anybody? What color did it become if it had the plant in it?
For me, it actually didn't become very blue. The starting solution, like when I like finished experiment, it was dark green, and so the one with the plant, it only got to like the pale yellow of yours.
Okay,
Yeah,
interesting.
sorry.
Say again,
mine was-
Bethany.
Mine was like a really pale blue as well at the end. But
So
it started
not
off pretty
as
pale blue.
bright. So there's a
Yeah.
couple reasons for that. What do you think reasons might be? Let's troubleshoot. Why might they not have turned a brighter blue?
Maybe lack of it being in the light, like in front of a window,
Yep. So how would that have affected it?
because the sun hitting the plant causes photosynthesis.
Right. So it's possible that they just didn't get enough light. Now, we did this later in the day for most of us, and it overnight may not have gotten enough light so that it didn't, natural light is always better. If you could put it in a window, you would notice a big difference. Now, full disclosure, when I did mine here, I had it under a grow light. So I put mine, I have a little hydroponic garden, and I put it beside my hydroponic garden. So it got that full spectrum light. Light bulbs just don't give you the same light that the sun does. So that's a possibility. There just wasn't enough photosynthesis to change it from that pale green, greenish yellow to blue. All right, what about the second one? What does the color change in the bromothymol blue solution indicate about the concentration of carbon dioxide in each setup? Well, let's use my results then, since you guys didn't really get major color change results. We had the color change. The color changes, remember why it's yellow? It's that yellowish green color because it has carbon dioxide in it. What does the fact that it became blue again at the end indicate about the carbon dioxide? There isn't any, or there isn't as much. Go ahead, Abigail.
I was going to say exactly what you were going to say.
Yeah, so the plant is using that carbon dioxide, taking carbon dioxide out of the water. So the plant uses the carbon dioxide and releases oxygen. So the color changes. So how does this experiment demonstrate photosynthesis? We kind of talked about that. It shows us that photosynthesis requires carbon dioxide and releases oxygen. All right, now let's compare the results in the dark condition. What did you observe in the one that was wrapped up in the foil? Bethany.
Mine remained pretty much unchanged.
Yeah. Yep. That's what we expected it to do, right? We expected it not to change because we weren't letting any light in. So no photosynthesis was happening. No carbon dioxide was being taken out of that solution. So that's what we would expect. And that is what we saw, unless anybody got different results. So now summarize the results of your experiment and explain whether your hypothesis was supported. So this is our hypothesis. If the plant is placed in a bromothymol blue solution and placed in the light, then the color of the solution will change from yellowish green to blue. Was our hypothesis supported? Bethany.
I would say that it was in most of our situations.
Yeah. And it did change a little bit, right? You had a little bit of a color change, maybe not as dramatic as if it had been in the sun. But yeah, I would say our hypothesis was, for the most part, supported. I think we would all have liked to have seen a lot brighter color, but that's okay. All right. Okay, now I want to do a case study about photosynthesis. And I want to remind you a couple things that you need to know in order to understand this case study. First of all, remember that the sun emits all this radiation, and radiation from the sun comes to us in waves. Waves that are really small, really close together are called gamma rays. And then they gradually get wider and wider and wider until we get all the way up to radio waves. So gamma rays are so small that they can do damage to living tissue. Gamma rays will destroy DNA and destroy structures inside the cell. They're so small. Radio waves are the size of a building. They're so big, they just go right over. There are radio waves flying around you all the time, but they're so big that they don't do any damage to living tissue at all. Right in the middle here of this electromagnetic spectrum of all the energy that comes from the sun, we have the wavelengths that we can see. So we can see ones between like 380. Oh, I'm sorry, I got a yawn. I'm trying to fight it, but it's going to take over. All right, hopefully that's it. So visible light is waves in between 380 and 750. That's what we can see. Now, different wavelengths of light are seen by the human eye as different colors. So a wavelength that's red has a different, a wavelength that we see as red has a different wavelength than what we see as yellow or green or blue or purple or any of the colors that come from the sun. Now, sunlight is white. Sunlight is white light, but it's actually a mixture of all those different wavelengths of light that come from the visible spectrum. When you mix all those colors together, believe it or not, you get white. Are any of you guys artists that mess with paint and colors? And yeah, so you know how, white is a combination of all the colors and black is an absence of all the colors. So photosynthetic plants, or plants that perform photosynthesis, gather energy from the sunlight. And they do it by using these light absorbing pigments like chlorophyll. Chlorophyll is the green pigment inside the chloroplast of most plants. And you guys learned last week that chlorophyll absorbs light very well in the red and the blue spectrum, but it doesn't absorb green light very well. Green light is reflected. So all the colors that come from the sun come and hit the leaf. They go through the clear outer part of the leaf and they go to the chloroplast. The green is reflected back. All the rest of them are used. So that's why the leaves of plants appear green, because it's reflecting back to us, because the color of light that you see is the color that is not absorbed. So green light from the spectrum isn't really used. The others are. Okay, so now we're going to do a little case study. This is Heidi. Heidi has become interested in gardening. She has planted a small potted garden in her dorm room, but she's distressed because her garden will not grow. She waters the plants every few days, just like the gardening books say. She's placed them near the window to get light, but her roommate, Mary, insists on keeping a translucent green curtain in the window. Mary wants to keep the curtain closed because she likes to sleep late, and she often forgets to open it when she leaves for class. Heidi hasn't made a big deal about it because it is translucent, meaning it lets some light shine through. One morning, Heidi asks Mary to stop at Pike's nursery to buy some fertilizer on her way home from work. Mary replies that she can save Heidi some money by getting some fertilizer from her dad's storage shed when she goes home the next weekend. Mary's dad is a corn farmer and has all the best plant products. When Mary returns home, she brings Heidi a bit of fertilizer in a bag labeled atrazine. After fertilizing the plants once, Heidi notices that the plants seem to be dying. The leaves are turning yellow around the edges. Finally, Heidi asks Mary to drive her to Pike's so that she can ask someone who works there about why her plants are dying. When she arrives at Pike's, Heidi begins talking to an employee named John. She tells him everything. When she finishes, John tries to hold back the laughter as he explains to Heidi why her plants are dying. All right, Bethany.
I actually had two things to say. Can
Okay.
chloroplast be any other colors besides green? Is that possible?
Well, so as the chlorophyll starts to die, remember that the other pigments that are in there, that are in the chloroplast, also
Mhm.
show up. So yes,
All right.
but not when the plant is producing chlorophyll. Chlorophyll is just such a powerful pigment that when it's there, that's what color the chloroplasts look.
Also, was it because her curtains were green?
Okay, so that's our first task, is to figure out what's happening. So task number 1, Heidi made 2 gardening mistakes here. Given what you're learning about photosynthesis, why weren't they growing and why are they now quickly dying? So let's start with the first one. What do you think there, Bethany? Give me more detail. Why does the green curtains matter?
Well, you said that green wasn't good, like green light is being filtered to the thing is not good for plants. So maybe she had a different colored curtain, it would work better.
Yeah, it isn't that it isn't bad for plants. It's just not as effective as the other colors.
Ineffectual is what I meant.
Yeah, there you go. There you go. That's a good word. So the curtains in the beginning, just the fact that there are curtains there means the light is being limited. Even if they weren't green, we would be cutting down on the amount of light that was getting to the plants. But the green changes that white light coming from the sun into green light as it shines through those green curtains. You can see here, this is the rate of photosynthesis under different lights. You can see that most photosynthesis occurs in this blue range and over here in the yellow-orange range. Very little photosynthesis happens under green light. So green light is absorbed least effectively by chlorophyll and it gives us the lowest rate of photosynthesis. So chlorophyll breaks down and leaves begin to turn yellow and die. Next question, why are the plants now dying quickly? So in order to answer this question, you have to know what this is. Abigail.
I was literally just gonna blame the plants dying on that.
I'm sorry, your microphone is real.
Can you hear me now?
Yeah, it's for some reason it's got a real like almost an echo. So speak slowly.
Okay, I was going to blame the plants dying on what's in that bag.
Yeah, so what do you think's in the bag? Somebody look it up. Somebody do a quick Google search. Look up atrazine or Atrex.
It's a product meant to cut down weeds.
Yeah, so it's a weed killer. It's called an herbicide. It's an herbicide. So here we go. Atrex, also known as atrazine, is an herbicide that kills plants rather than fertilizing them. The way it does that is by blocking the electron transport chains. You guys learned about cellular respiration. You learned that there are 3 parts to it. The first part is glycolysis, and it doesn't produce a whole lot of ATP. The second part is the Krebs cycle, and it doesn't make a whole lot of ATP. The third part, that's the electron transport chain. it produces most of, so like 2, 2, and 34, big difference. 34 ATPs are produced for every glucose in the electron transport chain. If you block that electron transport chain, now you're not getting ATP made in numbers big enough to support the life of the plant. So that flow of electrons can't be harnessed for ATP production. And without that ATP or adenosine triphosphate, the cells are not able to carry out life's processes and they will die, right? All right, so the next thing that we need to do is figure out what Heidi needs to do to fix the problem. So what do you think? Maria?
Maybe switching the fertilizer that she uses. Like get one that actually helps it grow.
Yeah, instead of a weed killer, it'd be nice if she actually used something that would help the plant instead of killing it. Yeah, good. Luke?
Maybe seeing what types of plants she has and applying the instructions and care for the plants she has to, like, basically giving the plants the right amount of water.
Yeah,
Because
give them
she
what
could,
they need.
maybe
Yeah.
she like randomly gave an outside plant, randomly put outside plants in her dorm room, which they're not meant to be there. So
Yeah,
that could have been a problem and
Yeah.
something she could check.
good, good. That's a possibility for sure. Different plants have different needs, and knowing what those needs are could help her to have a more successful garden. What else? Bethany?
Getting different curtains or just not using curtains like that.
Yes,
Keep the curtains
absolutely.
open.
Absolutely.
Keep the curtains open.
Yep. Keep the curtains open. All right. So putting the plants in the direct sunlight without any curtains at all would be helpful. Not using that atrazine would be really helpful. Using a fertilizer which promotes the growth of plants. Those are all things, basically giving the plant what it needs and not things that are going to kill it could be helpful. All right. So let's write a testable hypothesis for this. So remember what we mean when we're talking about hypothesis. We've got our if something is done, then something will happen. So we're going to be trying to change all of these things. So we're not really doing, we're doing a complex controlled experiment. It's not a simple one where there's just one variable that we're changing. So what are the things that we're changing here, and put them in the if part of the hypothesis? Abigail.
So the things that we're changing are putting it like changing how much sunlight, changing how much Atrax is in it, and changing the fertilizer.
Good. So now put that in if. Bethany.
If we put the plants in direct sunlight without Atrax and with a fertilizer, then it will promote plant growth.
Very good. Very good. So that's almost perfect. The only thing that's not perfect about that is we don't want to use we or I. We just want to say what needs to be done. So we use the passive voice in that. So we would say, if the plants are placed in direct sunlight and no atrazine is applied to them and a fertilizer that promotes the growth of plants is used, then the plants will recover and grow. That tells us what we're going to do in our experiment, the if part. We're going to put plants in direct sunlight without atrazine and with a good fertilizer. It tells us what we're going to measure. What are we going to look for? We're going to look for the plants to recover and grow. Excellent. Now, we're going to do all three of these things. And, in this case, I think we all know that they're all important things, but we could also do this individually. We could just simply put the plants in direct sunlight without any curtains, continue using atrazine and not use a fertilizer. The atrazine is going to kill them, so that's not going to be helpful. But we could do 3 separate controlled experiments, or we could do a more complex where we're changing all 3 of these things at the same time. All right. Anybody have any questions about the case study? All right. Here's some things that you really need to know about photosynthesis. So this is what's really important about it. First of all, it uses energy from the sun. It converts water that it gets from the soil and carbon dioxide that it gets from the air into glucose, which it stores for its own needs, and oxygen, which it releases and we benefit from that. We use it. Photosynthesis occurs inside the chloroplasts, which are the organelle in the plant cell that have the chlorophyll and the other pigments in it, and where photosynthesis happens. Plants need carbon dioxide, water, and sunlight in order to perform photosynthesis. If any one of the three of those is missing, photosynthesis will not happen. And a lot of energy is stored in one of those glucose molecules that is made by photosynthesis. There are two stages. There's the light reactions or the light dependent reactions. And then there's the Calvin cycle, which is sometimes called the dark cycle, sometimes called light independent. So that's what you really need to know. And you probably should know the chemical equation for photosynthesis. You can see here light energy is put in there. Sometimes it's put above that in an arrow. The other thing that's really important to know is that the products of photosynthesis become the reactants of cellular respiration. So what is made by the plant in photosynthesis is what we need in our mitochondria in order to keep making ATP, in order to make that adenosine triphosphate molecule. All right, so you did cellular respiration last week. I'm going to tell you, in my opinion, these are the two most challenging topics in all of biology. I think photosynthesis and cellular respiration are tough. They're tough to understand. So don't stress if you're finding this a little bit difficult. I think it gets easier from here. Genetics may be a little bit challenging too, but for the most part, this is the worst of it. Also the most fun to learn about. So they also are two processes that are critical for all of life on Earth. So all of life depends on these processes. Photosynthesis stores the energy in a glucose molecule, and all organisms need energy. They need that glucose to get the energy in it, but none of them, including the plant itself, can use that energy straight from the glucose molecule. They have to use this process called cellular respiration to get that energy out and to store it in a smaller molecule that is more readily available and easier for the cell to use. So when you take a look there at the word respiration, keep in mind, when cellular comes before it, it doesn't mean breathing. It's kind of really inconvenient that we use the same word for both. It does mean breathing, but when you put the word cellular in front of it, it means this process that makes ATP. So it is the total of all of the chemical and physical processes that organisms use to get oxygen to the cells and tissues and to get the products, carbon dioxide and water, which are given off. So when you remember that cellular respiration is a sum total of all those processes that get oxygen to the cells, you can understand that respiration is breathing and cellular respiration is what happens inside your cells. And this is the formula for cellular respiration. It's basically the opposite, the backwards of photosynthesis. Photosynthesis takes carbon dioxide and water and converts it into glucose and oxygen. Cellular respiration takes glucose and oxygen and converts it into carbon dioxide and water and gives off a lot of energy, these energy molecules. So just really think about all the things that you're doing as you're sitting here. Think about the things that you're doing voluntarily. You're hearing, you're thinking, maybe you're tapping your feet or tapping a pencil on your desk. Those are voluntary things. Now think about the things that you're doing involuntarily, that are just happening without you thinking about them. You're digesting your food. Your nerves are firing. You're breathing. Well, now that I've said breathing, you're probably doing that voluntarily too. Breathing is one of those processes that as soon as we think about it, now we're aware of it. Now it's a voluntary process. But when you're not thinking about it, it's happening involuntarily. Your heart is beating. Your blood's moving. Your bones are making blood cells. Your skin cells are undergoing division so that they can make new skin cells. There's all kinds of chemical reactions that are happening inside your cells, moving things in, moving things out. And all of those processes, everything that happens to keep you alive, requires energy in order to do its work. And that energy comes from the food that you eat. It comes from the food that you eat because the food that you eat has glucose in it. The glucose either was made by the plant or it was eaten by an animal that ate the plant. And then that glucose has to be opened up, Has to be broken apart into smaller molecules and converted into that energy storage molecule called ATP. What's ATP stand for? Bethany.
I just
So like I think adenosine phosphate, something like that.
Adenosine
Adenosine
triphosphate.
triphosphate.
Yeah, adenosine triphosphate. Okay, so you eat, you have your lunch. All that food has energy in it, and the energy has to get out of that food and into an ATP molecule, and it does it by a series of chemical reactions. Now it looks like just one reaction. It looks like, oh, it just takes it apart and puts it back together. It's actually a lot of reactions. I'll give you an example of that in a minute. But together, all of the steps that take us from glucose to ATP are called cellular respiration. And the final product, the thing that this whole thing is all about here, is ATP. So let's look at the reactants here. Remember, reactants react. They're what goes into an equation. So the reactants react. Look at the reactants of photosynthesis. So what are they? What are the reactants?
glucose and
And
oxygen.
yep, there you go. The most common sugar is glucose. So glucose is what is made by photosynthesis. Glucose can combine with other sugars to make other sugars, but we're focusing on glucose now. So what happens if there is no glucose going into the cells, into the mitochondria? Will this cellular respiration reaction happen? No,
No.
it will not. It will not happen. And if there's not enough oxygen, only a little bit of this first part, only the very first part of cellular respiration will happen. All right, now the products of cellular respiration, those are carbon dioxide and water. Carbon dioxide is toxic. It's a waste product that we have to get rid of quickly. How do we get rid of carbon dioxide? Maria?
By breathing it out.
Yeah, we breathe it out. So it's exchanged in our lungs. Our blood is filled with carbon dioxide. When it moves past our lungs, there's a lot more carbon dioxide in the blood than there is in the lungs. So it naturally just diffuses into your little air sacs in your lungs, and then you breathe it out. Easy as that. Very complicated process, but very easy at the same time. Okay, so you take a bite of your lunch. Here comes glucose now into your cells. Remember that the mitochondria are the energy producing organelles for the cell. So somehow we have to get that glucose into the mitochondria for cellular respiration to happen, for ATP to be made. Now, before it ever goes in to the mitochondria, a process happens that breaks that glucose molecule in half. That very first process is called glycolysis. So glyc refers to sugar and lyse refers to break. Glycolysis breaks the sugar. It breaks this glucose molecule into two smaller molecules. They're called pyruvate. Now, in the process, we've got 2 ATP that are made, but that's not really very much. We need a lot more than that. A really important thing that glycolysis does, in addition to those 2 pyruvates, is it adds a hydrogen to this NAD+. That NAD+ now is an electron carrier. It's going to hold the electrons until they can get into the mitochondria. So now, what happens to the 2 pyruvates and the 2 NADHs? That really depends on whether there's oxygen around or not. If you're running track and field event or running from a dog chasing you down the road and you're not breathing very well, there's not going to be a lot of oxygen present, but you still need to make ATP. So we have a process that allows us to do that. Anybody know what the process is that allows us to use, to go through glycolysis and produce a little bit of ATP? It's called fermentation. Fermentation. We'll talk about that in just a second. So glycolysis takes place out in the cytoplasm. It is not in the mitochondria. The very first part takes place out inside the cytoplasm. That is where glucose is broken down into 2 pyruvates and those 2 NADH. All right, now I told you guys that it looks like it's one chemical reaction, but it's actually a lot, a series. Remember that there are 3 parts of cellular respiration, there's glycolysis, the Krebs cycle, and electron transport. I'm going to show you what just glycolysis looks like right here. It's a little bit overwhelming, but I want you to see how complex this is. This is just glycolysis. This is just the first simplest stage of cellular respiration. And look at all these numbers here. Hexokinase. Number 2 is phosphoglucose isomerase, phosphofructokinase. Everything that's numbered here is an enzyme. Anything that ends in ASE is almost always an enzyme. Look how many enzymes are needed. Now, remember what an enzyme is? An enzyme is something that speeds up a chemical reaction, allows it to happen fast enough. If it wasn't for all those enzymes, they're proteins. If it wasn't for all those enzymes, you would not have enough. These reactions would not happen fast enough to give you the ATP that you need, even in an emergency situation when you're running from a dog. You got to have these enzymes. Okay, so here's what happens if there isn't any oxygen or enough. If there isn't enough oxygen present, we go through this process called fermentation. And really, this is just a matter of rearranging the atoms, kind of like you take apart a Lego structure and build a new one. Fermentation takes apart those 2 pyruvate or pyruvic acid molecules, and it rearranges them in order to produce 2 ethanol molecules. Now, it uses the electrons that were being carried by the NADH. It takes off these hydrogen and it becomes NAD plus again. And it releases 2 carbon dioxide atoms. Doesn't need them, releases them. And the 4 oxygen atoms that it doesn't need as well, that it doesn't use in the form of those carbon dioxide molecules. All right, now fermentation, that's what happens when you don't have enough oxygen. When you do, then those pyruvates go into the mitochondria. So this blue oval right here, that signifies this is inside the mitochondria. Here's our 2 pyruvate or pyruvic acid molecules. They go right on into the mitochondria, where they're immediately broken down into acetyl-CoA. Now, carbon dioxide is produced here too, and it's released. So when you've got this pyruvate that goes in, the total number of ATPs that are produced by this whole Krebs cycle right here, this part right here is 2. Once again, only 2. So we had 2 with glycolysis, we have 2 with the Krebs cycle, but that's not a lot. We still need a lot of energy we still could get a lot from that glucose molecule. It would be a waste to not use it. But we have this NAD that turned into NADH. This NAD turned into this NADH. This FAD turned into FADH2. This NAD here, this NAD here. We have a lot of these electron storing molecules. That's what is important for the third part of cellular respiration. This whole thing is called the Krebs cycle. From the Krebs cycle, they go into this electron transport chain. So those high energy electrons are going to pass from NADH and FADH2 to carriers that are in the membrane of the mitochondria. So in organisms that have mitochondria, this happens in the mitochondrial membrane. In organisms like bacteria that don't have mitochondria, remember they don't have organelles, this just happens in their cell membrane. Now, these electrons are moved from one protein in the membrane to another, to another, to another. And as they do that, as they move, energy from them moves hydrogen across the membrane into the space between the inner and the outer membrane. So that makes one side have a positive charge because all these H's are being pumped in that space. The H's are positive. The hydrogens have a positive charge. That gives this side a positive charge, and this side now has a negative charge. So they leave their energy inside the membrane. Well, now all these hydrogens start piling up here, and they got to get out, like you when you're in a crowded room and you feel like you just have to get out. Well, they have to have a way to get out. The only way that they can get out is through this molecule right here that's called ATP synthase. It is a protein embedded in the mitochondrial membrane. When those hydrogens move quickly through that ATP synthase, they cause the ATP synthase to spin like a water wheel. the energy that is used, that is generated by that spinning, is used to put this ADP and a phosphate back together into an ATP. This process right here makes between 32 and 34 ATP molecules from every glucose. So for a total of between 36 and 38 ATP molecules are formed from every glucose molecule. 2 of them are formed by glycolysis. That takes place out in the cytoplasm. 2 of them are formed by the Krebs cycle, and then around 34 are made by this electron transport chain. Now your book says 36. Most of the resources that I've seen say 38, but really doesn't matter. The bottom line is a lot. We get 4 ATP from glucose and citric acid together, 2 from each, and then 32 from this electron transport chain. Right? Pretty cool. Pretty cool. This is important. All right, so there's your quick review. Here's what you really need to know about cellular respiration. It takes 1 glucose molecule and turns it into 36 or 38 ATP molecules. There are 3 stages in cellular respiration. The first one is called glycolysis. It breaks down glucose. It lyses glucose into 2 pyruvates and 2 ATP molecules. The second one is the Krebs cycle. That is in the mitochondria. If there's energy, that those pyruvates go into the mitochondria, and it produces 4 NADH, 1 FADH, and 2 more ATPs. And then finally, at the end, we have the electron transport chain that uses the hydrogen atoms to produce 32 ATP molecules when those hydrogens whoosh back through that ATP synthase in order to cause it to spin and generate. Now, the most important reason that we need oxygen, that we breathe to get oxygen to our cells, is to safely remove the unwanted hydrogens at the end of the electron transport chain. So you got all these hydrogens. Let's take a look at these again right here. You've got all these hydrogens that rush through. Well, now you've got just a bunch of hydrogens sitting around that need to be removed because they still have some energy in them. A lot of it's been taken out, but they still have some. So we got to get rid of them. Well, what's the safest way to get rid of a hydrogen? Attach it to an oxygen. Now it becomes water. 2 hydrogens, 1 oxygen is a water molecule. Water is the safest molecule there is for the human body. So we have that oxygen that we need in order to get those unwanted hydrogen atoms away at the end of the electron transport chain. All right, anybody have any questions about cellular respiration? Are you still with me? You're all falling asleep.
I'm
As
with,
soon as I start feeling like I'm getting a little bit hoarse, I know I'm talking too much and you're probably falling asleep, but your eyes are all open, so that's good. All right, I want to just spend a minute talking about this molecule. So remember, ATP stands for adenosine triphosphate. And this is what it looks like. Each of these right here. What? Nope, that's not it. That's not what I want to do. I was able to get this to, no, I can't get it to stop. Yesterday I was able to get it to not freeze, but so each of these green balls in there is 1 phosphate molecule. There's 3 of them, adenosine triphosphate. So this colored, multicolored end right there, that's adenosine, and each of these is a phosphate molecule. You can think of ATP as like a battery for your cells. So it stores energy for you to do everything you want to do. And not just you, all living things on Earth use ATP. It's kind of like a tiny energy packet that cells use to get whatever they need to get done. So you eat, your body breaks down the glucose, it puts it into these ATP molecules, and then your body can use them. But how does the body get the ATP or the energy out of the ATP? So cellular respiration is all about making ATP. Now what? We still have to break the energy out of the ATP in order to use it. It does that with this very last phosphate at the end. That's the really important one. That last one right there, when it is broken off, it releases the energy that's needed. So your body makes ATP from the food that you eat. Inside your cells, the mitochondria break it down, break down the glucose, put it into ATP, and then the ATP goes wherever it's needed in the cell or outside of the cell in order to give you the currency, the energy currency that's needed by your body to be able to do the things that you do. So when an ATP reacts with water in a hydrolysis reaction, hydro means water, lysis means breaking down. So when an ATP molecule reacts, you end up with ADP, adenosine diphosphate, and one phosphate group and a lot of energy. So here you can see there's one phosphate. This is adenosine triphosphate here. You break this off, energy is released. ATP can be really thought of as the ultimate energy renewal source. Every one of your ATP molecules is recycled, goes from ATP to ADP to ATP to ADP, somewhere over 1,000 to 1,500 times every day. Think about that. Every molecule that's made. Basically, the human body turns over its own weight in ATP every single day. So if you weigh 100 pounds, your body produces 100 pounds of ATP over the course of a day. So the organelle that is responsible for the production of most of the ATP every human cell uses, what's the name of that organelle?
ADP.
No, the organelle inside the cell. Remember, chloroplasts are the organelle that make glucose. What's the one that breaks glucose down into ATP? The energy producer of the cell. Maria.
Mitochondria?
Yeah, the mighty mitochondria. That's the way I remember it. Picture a superhero. The mighty mitochondria making the energy for the cells. Right. All right. So here's a question for you. Why use ATP at all? Why not just break open that glucose molecule and let the energy out? Why use wires in your home? Why not just have all the electricity just flying around?
Because you'll get electrocuted.
Yeah, you would get electrocuted. Exactly. A kind of a similar thing would happen if we just released all that free energy from a glucose molecule into the cell. It would actually result in too much thermal motion. So thermal motion is the movement of the particles things are made of. And there would be too much of that. It would damage and then eventually destroy the cell. So a cell has to have a way to handle the energy in a way that lets us store it safely and release it only as it's needed. So we do that by making ATP. Pretty cool. Just like a rechargeable battery. And then how does the cell get the energy out of ATP? It does that with enzymes. How does ADP become ATP again? It does that with enzymes. Enzymes are pretty important. So that's we're going to probably end with this video that shows you how ATP goes back and forth between ATP and ADP.
Almost everything an organism does requires energy. But where does this energy come from? To answer this question, let's zoom in on one of this hummingbird's cells. Notice that each cell contains many mitochondria, which are often referred to as powerhouses of the cell. This is because mitochondria are responsible for producing most of the cell's energy in the form of ATP. ATP, or adenosine triphosphate, is really important because it serves as an energy carrier in cells. It's responsible for moving the energy found in chemical bonds of our foods to the areas of the cell that need energy. Without a constant supply of ATP, cells would quickly die. Let's zoom in to take a closer look at ATP's components. ATP is composed of a nitrogenous base called adenine bound to the sugar ribose and 3 phosphate groups. The bonds between the phosphate groups are high energy. An enormous amount of energy is packed into these bonds. When energy is needed, the terminal phosphate group is removed in a process called hydrolysis, releasing energy and forming adenosine diphosphate. The energy released from this reaction is used by the cell to carry out all the functions necessary to sustain life. Later, at the mitochondrial level, ADP and phosphate are recombined back into ATP, recharging the energy pathways of the cell. This cycle from ATP to ADP and back again to ATP is called the ATP cycle.
Okay, the only thing I really don't like about that video is it shows donuts at the beginning for food. Donuts are not food. They do give you some glucose, that's for sure, but don't consider donuts food. All right. Anybody have any questions about anything that we covered today? All right. You are free to go. Thank you all for coming, and I hope you have a wonderful week.
Have a
See you
nice
next
day.
time. Thank you.
Thank you.